Hi everyone. So today we are going to focus on intracellular receptors. But even before we start with what is intracellular receptors, let's try to understand the basic knowledge of what are receptors. The ideology of receptor is pretty simple. It functions in a way that receptors generally look out for ligands. These are small molecules, okay, which when attaches to a particular receptor, it will start a cascade of events. This cascade of events leads to a sequence of changes, or we can say alterations, in the receptors, which further transduce the signals required for performing any kind of cellular functions. So that's the basic idea of a receptor, okay? Something binds to it and it starts acting like a messenger, or at least activates some other messenger. We have all these same receptors like this, okay, in this particular diagrammatic format. But receptors are not always just of one particular structure. There are multiple shapes, sizes, binding sites of receptors. And depending upon their shape, size, and even the recepting structure, ligands are also different. Okay, so let's dive on to today's topic. The major concept, types of receptors. Now, generally, there are two types of receptors in one, as we can see in structure one. A ligand is coming and binding to the receptor. That's common. That's common for the receptors to get the ligand. Only difference over here is the location. If you compare the two diagrams, you will definitely get to know that in one particular diagram, the receptor is on the cell. You can see over here, it is on the membrane. It is attached to the membrane. Whereas in the second diagram, you can find the receptor to be inside of the cell. Okay, now this is the major difference of receptors. That is, types of receptors based on location. Function is the same. Function is to catch a ligand and then keep on transducing signals inside the cell. Function remains the same. What is the difference? It's the location of the receptor. So in the first diagram, as you can see, they are basically cell surface receptors. So they will be definitely present on the surface of the cell, not inside the cell. Okay, and the second one are intracellular receptors, which we even commonly call as nuclear receptor. Apart from the location of these receptors, the major difference that arises is the kind of molecule that they bind with, that is the ligand. So even based on ligand, the location can sometimes be described. What happens is ligands generally, like a good chunk of them, are hydrophilic. And some of them are hydrophobic. Now, those which are hydrophilic, they cannot pass through this plasma membrane. As we all know, that passing through the plasma membrane, a particular substance needs to be lipophilic or hydrophobic. So hydrophilic ligands cannot pass through the plasma membrane and definitely cannot enter the cell. Now, if they do not enter the cell, then how will they carry on their own function? That is the reason why the receptors for those hydrophilic ligands are not in the interior of the cell, but rather on the exterior, that is on the surface of the cell. So as you can see that, depending upon the ligand type also, sometimes the receptor location might be very crucial. In the second diagram, you can see that the ligand molecule is passing through this plasma membrane and entering inside the cell. And as I just mentioned, that for a particular molecule to enter the cell, it requires to have some type of hydrophobicity. So this ligand basically is hydrophobic, and that's the reason why it can pass through this particular plasma membrane. For such ligands, generally the receptors are internal to the cell or inside the cell. That's why the term comes intracellular receptor. Comparing these two types, major functions and ideologies of how these receptors are going to transduce the signals can take place. Today, we are going to focus more on intracellular receptor. Now, intracellular receptors basically are of three types. They could be receptors. They could be receptors for steroid hormones. They could be receptors for non-steroid hormones, or they could be orphan receptors. Okay, so depending on the ligand, the receptor type might change. Like with the term steroid, we all know that steroid hormones are the ones which are generally derived from cholesterol or fat. Okay, so definitely they can be internalized. To give some examples for this, I think we can aim for such things like progesterone, which is a good steroid hormone that we can think of, estrogen. Okay, you have got others like androgens, the other androgens which might be present. Along with that, we can even look for corticosteroids, which are steroid hormones, and even certain vitamins like vitamin D. Now, for these hormones or these particular ligands, the receptors are generally present within the cell. That is either in the cytoplasm or in the nucleus. So basically 90% the internal receptors are for steroid hormones. A small chunk of it is also present for non-steroid hormones, very less. The reason being non-steroid hormones mostly are derived from peptides or proteins or amino acids. So their hydrophobicity is basically dependent on the amino acid sequence. If the amino acid chain is hydrophobic in nature to a good extent, then definitely these hormones can pass through the cell. But a good chunk, when they are not hydrophobic or hydrophilic, they won't be able to pass. So very few examples exist for non-steroid hormones, mostly of which you can find, you can say vitamin A or retinoic acid. Along with that, one major category is thyroid hormones. Okay, so they are non-steroidic in nature, but yes, because of a good chunk of them being hydrophobic, that's the reason why they can pass through the membrane. Now coming to the question, what are these orphan things or orphan receptors? Now there are two types. There are orphan ligands and orphan receptors. Orphan ligands are the ones whose receptors have not yet been discovered, whereas orphan receptors are the ones whose ligands are yet undiscovered. Okay, so depending on whenever the discovery takes place, the orphan receptor, orphan ligand definitely gets up a name and they get categorized in the following. Now that we have understood receptors, types of receptors, and even the types of nuclear receptors or intracellular receptors, there's a specific way or a structure in which a nuclear receptor is designed. Let us look at that in detail. Okay, so we'll be studying the structure in detail. Please pay attention on it. Now, when we look at this particular sequence, our nuclear receptor basically has an N-terminal and then there is a C-terminal to it. And there are various regions according to which it is classified. So we'll first look at the regions. Okay, there is a AB region. Then we have got a C region. There is a D region. Then we have got an E region and F region, of which the F region is not yet been perfectly categorized or classified. So that's the reason why many discoveries are yet going on the C-terminal F region. So what are these regions basically? So to understand them, I think we should first look at their names. Okay. So in the N terminal, at the N terminal, the AB region basically signifies something called as AF1. It's called as activation function 1 or transcription activator function 1. C region generally is called as DBD, that is DNA binding domain. Okay, so basically this portion of the receptor is going to go and bind to the regulatory gene. The D region, on the other hand, is called as a hinge region, and not much is being discovered of this part. E region is divided into two. So basically it is LBD, that is ligand binding domain. Over here, the ligand is going to come and bind. And there is another AF2, that is activation function 2. Okay. So these are the domains, or these are the regions that we are going to look for in the structure. Okay, so what is going to happen in this AF1 and AF2, I'll be discussing in a minute. DBD, let us try to discuss first. So DBD is the region where your receptor is going to go and bind to the DNA. LBD is generally the region where your ligand is going to bind. So ligand binding site is going to be there at LBD. Okay, now coming to the major question. What happens at AF1 and what happens at AF2? Okay, AF1 and AF2 both called as activation domains. Okay, so AF1 is basically ligand independent activation domain and AF2 is ligand dependent activation domain. Why is the difference over here? We can see LBD is ligand binding domain. So that's the reason why whatever functions that need to be activated, okay, those functions, those co-regulatory molecules will be activated or recruited by AF2. Okay, so it is very much dependent on the binding of the ligand. AF1 has a regulatory function. Okay, it is not activated by any ligand. It is independent of any ligand. Okay, that's the reason why it is not present at the LBD region. AF1, on the other hand, is recognized by many regulatory molecules, and that is the reason why it is going to function properly. Okay, so if I have to talk about these regions, we have got 1, 2, 3, 4, 5 to 6 regions in the nuclear receptor structure, namely A, B, C, D, E, F, of which F has yet to be classified and characterized. There's more study going on it. So up till then, I think we can keep F by the side. AB region towards the N domain, that is N terminal, we have got transcription activation domain 1, which is ligand independent. Then we have C region, which is DBD, DNA binding domain. Over here, this is the particular region or sequence which will go and bind to the DNA. Okay, we all know that receptors definitely transduce the signal. So one of the way in which they can transduce is by activating or binding to the gene regulator. Okay, so that is the way how DBD is coming into picture. Hinge region. Then we have LBD in the E region. LBD is ligand binding domain, where ligand is going to come and bind. So this particular structure of the nuclear receptor is very specific for the ligand. As we all know that receptors are always specific for their ligands. So only specific ligands meant for this receptor are going to come and bind over here. Okay? And then we have got AF2, which is ligand dependent activation domain. So this will get activated and will start recruiting other regulatory molecules only when the ligand binds. And that terminates with a C terminal. I hope you could understand this particular topic. Thank you.